A very good morning. My name is SAMEEKSHA Chauhan and my topic is galactose operon. Galactose operon is also known to be an inducible pathway, which means that the expression of genes involved in galactose metabolism is turned on in response to the presence of galactose in environment. So when we grow yeast, which is an eukaryote, and the example is Saccharomyces cerevisiae, where we see the galactose operon. Then we have to add galactose so that this galactose operon can function. Here in the image, we see three genes called GAL3 gene, GAL80 gene, and GAL4 gene. Um, um, I was talking about these gal genes because their interplay is what the whole galactose operon is about. Their interplay in the presence and absence of galactose makes a lot of difference. Galactose operon is different from lactose operon because it's not a operon. It is just the gal genes that are clustered, and they are together because they have similar promoters. They are regulated coordinately by a common set of proteins. We have important players here called gal genes, data box, which is our promoter, INR sequences, and upstream activator sequence. Upstream activator sequence is an enhancer sequence which is recognized by our main player, which is DNA binding transcriptional activator, that is our GAL4 protein. Now, the genes that I have mentioned over here in the image encodes for the respective protein. GAL3 encodes for GAL3 protein and the other respectively. We'll be studying about them a little later, but before that, we'll just look into how the pathway of converting D-galactose to D-glucose 6-phosphate has to occur so that it gets into the glycolytic pathway and then the yeast can utilize it. The gene over here, even in this image, GAL7, GAL10, and GAL1, that encodes for these enzymes so that this conversion can be possible. Moving ahead, that if we study the whole interplay of these proteins that I have mentioned, 84 and 3, in the absence of galactose, then it looks like this. Starting with our main player GAL4P, it is always in the dimer form, so it attaches to the USG, which is our upstream activator sequence, within the dimer form, and it always have an activation domain. Now, this activation domain, when the galactose is absent, is occupied by our GAL80P protein. Here, we don't want to transcribe gal gene because, yeah, of course, because galactose is not present. So here what happens, GAL80 inhibit the GAL4P by binding to its activation domain and preventing it from activating the nearby gal genes, and thus transcription cannot occur, and further the cycle remains into the rest state. But what if the galactose is present? This is a image. Apart from this, I have a better image over here where galactose then interacts with our GAL3P protein, which is always there in the system, but its importance occurs when the galactose is present. This interaction, or say complex formation, results into the conformational change, or say allow the GAL3 protein to attain the ability to have high affinity towards the GAL80 protein, and now it drives away this GAL80P protein so that our GAL4P protein is now free to carry on, or say activate the other GAL genes and start with the transcription. Moving ahead, how this activation occurs, this is done in 3 steps. The transcription activator is TF2D, how we had cap in our lactose operon. In a similar manner, TBP is just a part of TF2D, which enhances the complex formation between the GAL4 protein and TF2D. The TBP main purpose is this, and then this together, the GALP protein then allows the binding of this TF2D to Tata box, which is our promoter. In the similar manner, our second point is mediator complex. Mediator complex is basically complex these are such certain proteins that makes the accessibility of RNA polymerase 2 um, and helps the complex formation between Cal4P protein and RNA polymerase 2, so that now this RNA polymerase 2 can go and bind to the promoter. Before this, the promoters always have nucleosomes over them, and that's why recruiting of SWI-F slash SNF is necessary, so that they move the nucleosome by remodeling them and then emptying the space over here so that these two complex TF2D and RNA polymerase 2 can bind over it and can start with the transcription. But what happens when galactose is present along with glucose? Always glucose is favored over lactose, galactose, or anything that we have already studied in lactose operon. So here also, when glucose concentration increases, the MIG1 over here accumulates, and then they recruit the tube1 over here. Tube1 is nothing but a co-repressor called HDAC, which is histone deacetylase, that remove acetylase group from nucleosome and then results the chromatin to be tightly packed. That tends to have now inactive gene. So here the GAL1 is being repressed because of the HDASE, which results in tight chromatin conformation and recruiting more of the inactive gene. And now the glucose is being utilized. So moreover, galactose operon is this that I have discussed. And so yeah, thank you.